not a tiny group. So it's my pleasure to introduce Rachel, who earned her PhD in infectious disease immunology at UCLA and performed postdoc research in HIV virology and immunology in Alberto's lab at GW. She's currently completing a 2-year NIH Fogarty Global Health Research Fellowship in Lusaka, Zambia. She completes it in March through the University of North Carolina, Chapel Hill, and she's currently investigating immunological mechanisms of preterm birth in women with HIV and contributing to multinational studies of anemia and pregnancy. Additionally, she is an MPH candidate at Hopkins with a focus on epidemiology and reproductive health. Rachel, why don't you try and pull up those slides and we'll get started. Thank you so much for being here. We're thrilled to have your presentation. We're going to have people turn off cameras just because Rachel is currently in Zambia and we're a little worried about bandwidth. So for the slide presentation, we'll turn off cameras and then we'll turn them back on for discussion. Rachel, we currently see like your slides, but they're not in presenter view yet.
Got it. I just wanted to say hi to everyone, and now I'll also
turn
off my I'll turn off my camera. Thank you all for being here. Let me just do that. And everyone can still hear me, right? Because like the little bar disappears. Yeah. So I just want to make sure.
I can hear you. I won't take myself off mute until until your slides are up and we're sure that all is well. Okay, perfect.
Okay. I can't see any of you at the same time, so I'm just going to I'm just going to talk to my screen, but I really appreciate the opportunity to be here today, and I've really enjoyed keeping in touch with everyone in the DC CFAR. I hope I have this all of the scientific interest groups listed correctly here. I know this is a joint effort, and again, I appreciate the chance to be here. Please bear with me if at any point I'm searching for words. I'm really jet lagged right now. I just returned to Lusaka a few days ago, so I'm still kind of catching up. But what I thought I would do is present sort of a 2-chapter presentation. It should be just a little more than maybe 45 minutes or so. I hope that's okay. You know, if I start going over at the end, we can just kind of wrap it up. But I thought I would present sort of a chapter from my postdoc research with Alberto, who I appreciate coming here today. And then the second chapter from my current Fogarty Global Health Research Fellowship work, which is again here in Lusaka, Zambia. So the first half is a little bit more basic biomedical science. So I know we have some clinicians in the audience. So I will do my best to just explain it in a very straightforward manner, and feel free to take a coffee break and come back about halfway through when we get to the more clinical aspects of the presentation. I won't be offended, and I can't see anybody anyway. So again, so I've sort of been working on this signaling pathway, sphingosine 1-phosphate or S1P for quite a while now, since my PhD training in the lab of Crystal Bohart at UCLA. And when I transitioned to postdoc research with Alberto, I was really interested in kind of continuing my work on that pathway, but I had only started looking at HIV toward the end of my PhD, and I had really become fascinated with HIV research. I wanted to continue along that path, and I wanted to continue looking at S1P, and Alberto was open to that. So I'm really excited to present a little bit of the work we did together on that. And then currently, I'm still looking at S1P, but in a very different context. This is in more of a clinical setting here in Zambia in the female reproductive tract, and still working with HIV as well. So it's been a bit of a 3-chapter project spanning several years, and I'm happy to present some of that today. So I'll give a very basic background into S1P, or sphingosine 1-phosphate. It is a signaling sphingolipid molecule, or a lysosphingolipid. It's expressed in most cells and tissues in the body, so it's ubiquitous. And there are 5 7-transmembrane receptors. If you can see the pointer, this is just a visual of that, but no need to focus on the minute details here. I just wanted to show there are 5 characterized receptors and 2 kinases, sphingosine kinase 1 and 2, which are responsible for generating S1P from phosphorylation of sphingosine, a precursor. There are various downstream signaling effects, and those are really mediated by the differential binding of S1P to one of the 5 different receptors, which are expressed differentially. So, for example, when binding to S1PR1, then you may have immune trafficking effects, different cell development effects. But with S1PR5, there are very different effects on NK cells and the CNS, for example. And then the clinicians may be familiar with some of the inhibitors that are actually used clinically, for example, fingolimod, which I'll be talking more about, or FTY720, is an inhibitor or a modulator of 4 of the 5 S1P receptors. And you can see that indicated here. So it actually modulates their expression, and clinically it's used for multiple sclerosis, and it really just mediates some of that immune trafficking, influx of immune cells that are responsible for some of the symptoms. So that's really all you need to know to understand the S1P background for the presentation. Again, I mentioned I won't be talking about this in this talk, but I've previously worked in the context of S1P signaling in the human thymus. And we saw that in HIV-1 infection of the human thymus, there's an increase in the expression of one of the receptors, S1P receptor 1. And we saw that this was mediated by cytokine fluctuations secondary to HIV infection, namely interferons, TNF-alpha, and that upregulated S1P receptor 1. And we think there are possible implications for T-cell reconstitution, for example, in individuals who are adherent to ART but do not adequately reconstitute CD4 levels. Then in Alberto's lab, I was really interested in looking at this from the other end of the other side of the relationship. So instead of looking at the effect of HIV on S1P, I wanted to see whether we could target aspects of S1P signaling to inhibit the progression of HIV infection. So what we did was we looked at targeting both the receptors for S1P and the kinases. I started with the receptors during the first couple of years, and then in the second half of my postdoc, we looked at the kinases. And we saw that if we target different aspects of S1P signaling, we can inhibit the progression of HIV infection in primary CD4 cells. And again, no need to focus on all of the details here, but I'll talk a little bit about this in the coming slides. And then when I started my Fogarty Global Health Research Fellowship in 2022, I was still interested in S1P and still interested in HIV, but I really wanted to get some experience in translational research. So I proposed a project where I wanted to look at, first of all, whether we could detect S1P signaling in the female reproductive tract, and then whether we saw any changes with HIV and possibly with pregnancy outcomes, since I'm working here with a group that has a long-standing longitudinal antenatal cohort. So in the first part of the talk, I'd like to talk about targeting S1P signaling and HIV infection. So many of you, because Alberto is such an involved member of everything in DC and in the CFAR, have probably seen this this primary cell model of HIV latency presented before in various contexts. But I'd just like to give another overview before we get started with the data. So what I'm presenting to you is really 2 different versions of this model that I used during my time in the lab. And this is a shorter version of the model, which we termed cell-free infection for the experiments that I was doing, and then a slightly longer version, which we termed cell-to-cell infection. So the whole general picture of the model is that it is a primary T-cell model of HIV infection, and it starts with isolation of naive CD4 T cells from relatively healthy human PBMC donors. We activate them with anti-CD328 in non-polarizing conditions, so we block the cytokines that promote T-cell differentiation to various pathways, and then we expand in the context of the cytokine IL-2. I'm indicating here that during this time period, we can pre-treat with some of these modulators that I mentioned, like FTY720, and then we infect at day 7 of the model with, I'm indicating here NL43, this is an X4 tropic HIV virus, or NL88, but we've also used various primary isolates, different molecular clones, and transmitted founder viruses in this model. So we infect for 3 days, and then in this somewhat shorter version of the model, we can assess HIV intracellular P24 at day 10 by flow cytometry. Then in some of the other experiments I'll show you, we did a slightly longer version where we wanted to infect at day 7, keeping everything else the same, and then crowd the cells in round bottom plates for 3 days. This facilitates cell-to-cell transmission because the cells are close together. And with this version, we were interested in looking at what if we treat with these modulators to S1P receptors during the cell-to-cell transmission. So we're treating at a different time period. And then after 3 days, again, assessing HIV P24 by flow cytometry. So what does this look like when we treat with these S1P receptor inhibitors in the model? Well, this is just showing a flow cytometry plot, and I've arranged them all pretty much the same way. So this is CD4 expression on the Y and HIV P24 on the X. So this is what the uninfected cells look like. Some of you are extremely familiar with flow, some of you may not be. So just, you know, it's overly simplistic perhaps, but you can see they have a lot of CD4. This is what we expect, and they're uninfected. They have no HIV protein. This is what the infected cells look like at day 10. This is a pretty normal percentage of P24 expression. And you can see that there's a dose-dependent response to treating with this F2Y720 with this S1P receptor modulator from 30 nanomolar up to 100 nanomolar of F2Y720. And you can see that the HIV P24 decreases with increasing F2Y720. I see there's a comment in the chat, so, oh, hi, Martha. Oh, thank you, nice to see you. So again, you can see a dose-dependent decrease in HIV P24. And this is just summarizing that for 2 different HIV molecular clones that we used, NL43 and NL88. So these are different molecular clones that utilize the CXCR4 and CCR5 co-receptors. So that's just the main difference between what you're seeing here. And for both of them, irrespective of the co-receptor utilized, you see a dose-dependent decrease in HIV P24 with F2Y720 in increasing concentrations. This is just showing the curves for both of those, the summary of both of those plots overlaid. So you can see that with NL43, there's a little bit different of a shape. It starts out maybe a little bit steeper and then kind of a somewhat greater effect with the higher concentrations. We do also sometimes see slightly higher infection with NL43, but then again, I had this, this donor here with NL88 with quite high infection. So it really just depends. A lot of donor variability with human cells. So then we were curious, what if we treat during this cell-to-cell transmission stage? So once the cells are already infected and then crowded close together. So what I'm showing you here is uninfected, HIV infected, and a medium to high concentration of F2Y720. So in different experiments, we just optimized the concentration of this modulator at about 60 nanomolar, which was effective, but there was no effect on the viability of the cells. A little bit higher, above 100 nanomolar, I started seeing that there was some cell death. And so, of course, if we kill the cells, there will be less infected. So we didn't want to use those higher concentrations. So you can see that with this concentration of F2Y720, there's about half the HIV P24 as compared to the untreated. And this is just a summary of that, showing F2Y720 versus untreated for several different donors. Again, the same axes. This is the P24 level. So I mentioned at the beginning that this is a primary cell model of HIV latency, and that may have been what you've seen in previous presentations from Alberto's group, because this is really what he's known for. So this is really exciting because we can look not only at the effect of productive infection, which is during these earlier stages, cell-to-cell transmission around this time period, but also we can look at the establishment of latent virus. So we were then really curious, well, if we treat during this cell-to-cell transmission stage with the S1P receptor modulator, does that correspond to an effect on the latent virus that has been established and that we reactivate to check later on? So you're probably all familiar, but just for those who may be in a different field, one of the main challenges in working with HIV is the ability of the virus to establish latency. So it can enter cells, and then those cells may go, it integrates its material into our genome, and then they can go into sort of a resting state. So a person may be seemingly fine, and then in the future, if those cells are reactivated, they can begin to produce virus again, and again, there can be viral levels in that individuals. And this is one of the main reasons why looking into a cure for HIV is so challenging. So we were interested in looking at whether treating during cell-to-cell transmission would correspond to a difference in the reactivated latent virus that may have been established. And we've previously seen that the establishment of latency is really closely related to the infection that occurs during this cell-to-cell transmission stage. So we thought that that would be a likely extension of what we had seen. So the model is essentially the same up until the day 13 point that I described earlier. Only instead of stopping at this point and assessing the P24 level, we uncrowd the cells, treat with antiretrovirals, and then culture for another 4 days. So what happens during this time is that the productive infection is decreased, but some of these cells during this time frame from days 13 to 17 may have been latently infected. So they may be latently infected. So we reduce the level of the productive infection, but again, some of the cells we're interested in those because they may not be productively infected, but they may have latent virus inside. So at day 17, we isolate the CD4 positive cells. So these are the non-productively infected cells, and that's because productively infected cells downregulate CD4 expression. So once we do this isolation, we have cells that may have been latently infected. We then do a reactivation, and at day 19, we assess any virus that may have been produced following the reactivation. So I just want to focus your attention here on the reactivation only. That's with anti-CD328 beads, and then the reactivation plus the FTY720. And again, this treatment was done way back during day 10 to 13. So we wanted to see if any effect we caused during that time is apparent later on with the reactivation. And it's a little bit hard to see a striking effect because of the scale and this one outlier and everything, but we did see a decrease in the latent virus that was reactivated at day 19 when we had treated with FTY720 much earlier in the model. So essentially, we did see that reduced transmission during this time directly corresponded to reduced reactivated latent virus in CD4 cells. And with that, I'd just like to share a few slides showing some of the mechanisms that we looked into. This is by no means exhaustive, but I wanted to try to keep this for a general audience. And if you're not a basic scientist, I really appreciate you bearing with me this far. So I just have a few more slides of this before the clinical research. So we were really curious what might be the mechanism of what we were seeing. And the first thought that we had was there may be a reduction in essentially the total HIV DNA and or the integrated HIV DNA in cells. So in collaboration with Dr. Chamont and Remy up in Montreal, we sent samples that were either untreated or FTY720 treated, and they performed their well-established nested PCR assay for total and integrated viral DNA. And this is expressed as copies of HIV DNA per 10 to the third CD3 cells. And what we saw was when comparing the FTY720 treated to the untreated, there was a reduction in the total HIV DNA, as well as in the integrated HIV DNA in cells. So we did in fact see an effect at both of those stages. In agreement with that, when we looked by flow cytometry at CD4 levels on cells, so that's the main receptor that the virus needs to enter cells, we saw with FTY720, there was a reduction in the CD4 density as evaluated by the mean fluorescence intensity of CD4 by flow cytometry. And in keeping with that, we also saw that there was a reduced relative binding, although again, a lot of donor variability, but overall across the board, there was a reduction in binding. And we just assayed this by a very simple experiment where we incubated cells with virus for a short time and then lysed the cells and looked by P24 ELISA. And then in agreement with that, I'm not showing this data here, but we worked with the lab of Dr. Bukrinsky and Larissa from his lab. And we saw by a viral fusion assay that there was also reduced viral fusion with FTY720, which makes sense since we had seen reduced binding and reduced CD4 density. So then lastly, or at least lastly for this presentation, we looked at the effect on the innate antiviral restriction factor SAMHD1. which controls the virus by restricting dNTPs needed by the virus. So we looked at whether there may be an effect on SAMHD1, which could be contributing to what we had seen, along with the other mechanisms pertaining to binding, fusion, and CD4. And we saw that when treated with FTY-720, there was a reduction in the inactive form of SAMHD1, which is the phosphorylated form, so pSAMHD1. So reduced pSAMHD1, but no real effect on the total SAMHD1 levels. And this is just showing two representative donors for the inactive SAMHD1 and the total SAMHD1. So we think that that was probably contributing, at least to a certain extent, to what we had seen with the binding, fusion, and other mechanisms. But there was increased activity of this antiviral factor. So just to sum up what I've shown you for the more basic science side of things, we think that by targeting S1P signaling, we can inhibit progression of HIV infection at various stages of the viral life cycle, including binding and fusion, CD4 levels, and SAMHD1 activity, which affects reverse transcription and integration, which is what we had seen with the total and integrated virus levels. We think, I'm not showing you that data, but we think that may be tied to cyclin D3 and possibly other cell cycle components. And overall, at various stages, there seems to be an effect of inhibiting S1P signaling. So something we were really curious about, but unfortunately I don't have time to show it today, is what about the effect on the targeting the sphingosine kinases? So we were also really curious if we targeted sphingosine kinase 1 and 2, which I mentioned generate S1P itself. We were curious if that would also have a similar effect. And I won't present it, but just to sum it up, we did those experiments and we saw that if we target sphingosine kinase 1 and 2, we do see an effect by using DMS, which is a dual inhibitor of those kinases. And we also saw an effect just targeting sphingosine kinase 1. So overall, DMS, SKI2, which is the kinase 1 inhibitor, and FTY-720, so 3 different inhibitors targeting different aspects of S1P signaling, inhibit productive infection and latency in various models. They target S1P receptors and the kinases, again, and they seem to reduce phosphorylated or inactive SAMHD1 levels, which contributes to the overall effect and some of the other mechanisms I've shown as well. There are different effects with pre-treatment, like I showed you at day 5 before infection versus treating the total cells, like I showed with the cell-to-cell at the later time point. And we also saw that sphingosine kinase 2 inhibition is really not as effective and has effects on viability. So if you're interested, there are a couple of papers that I've listed here. But at this point, having given that very extensive background about the basic biomedical science, I'd like to pivot the conversation and spend the rest of the time on my current global health work here in Zambia. So everyone can come back now from your breaks. So I'd like to start with a few photos of my time here in Zambia. This is the lab that I'm currently working in. It's a small container actually converted into a lab in a really ingenious way. And we have, you can't see it because we're sitting right in front of the whole workflow here, but the samples come in sort of in front of the lab, which is where my pointer is, and then move through this whole workflow where we do 10, 20 different tests, depending on what study we're working with. There's CD4 levels, electrolytes, kidney and liver function, hormone like thyroid testing. And then in the back, we even do have a flow cytometer. And I haven't been lucky enough to use it because my work has been PCR based, but it's really an efficient, very tight space. In fact, working there, it very frequently feels just like this photo with everybody sort of on top of each other, but it's been a really great experience. And it really makes you appreciate the whole concept of space that in the US, everything seems so big now when I come back in the labs, but it's been a really great experience and a really welcoming group so far. This is Humphrey, the head of the lab, and this is Gabby, who you also see here, along with a colleague in Lilongwe. This is when we took a work trip to Malawi one of our neighboring countries, to work with the lab there. And it was a really exciting experience. They were actually having a cholera outbreak at the same time. So I got to shadow some really high stakes and emotional things while I was there. So that was really a great experience. And then this is just the view outside of the lab. These are two kids who really like to hang out there. And I guess they made some costumes out of boxes, which I thought was quite ingenious, and they look really cute. And I think they're going to pop up on one more slide toward the end. We actually currently have some visitors from GW with us right now. They're just about to leave tomorrow morning. This is Dr. Moores and Abby. And this was just a couple of days ago with some colleagues, and we were having lunch with them. And then these are two of the ladies who I was working with at one of the trainings that we did when we expanded one of our protocols, and just gives you an idea of some of the vibrancy of Zambia, which I really love. So anyway, with that background, I'm currently appointed with the University of North Carolina at Chapel Hill, so UNC, and my funding is from the Fogarty International Center at the NIH, with co-funding by NICHD, which I'm very grateful for. And when I entered this fellowship, as I mentioned before, I felt that I had the solid biomedical background, and I was really interested in getting some experience, either for the couple of years or as a career transition. Either way, I was excited to get some experience in clinical translational work. And so I had the idea, first of all, to look at whether we could even detect S1P signaling in the female reproductive tract. As I mentioned, I'm working with a group with a lot of experience in women's health and pregnancy, and we have a long-standing longitudinal antenatal cohort based here in Lusaka. So I was lucky enough to come into that established study environment and to have access to specimens, vaginal swabs that I could work with. So that was my first question. Could we even detect S1P signaling? And then did we see any changes in the signaling pathway with HIV infection or with preterm birth? So I'd like to give a little bit of background about HIV and pregnancy in Zambia. So this is actually a global phenomenon. This first point not only doesn't only pertain to Zambia, but women living with HIV have approximately twice the rate of spontaneous preterm birth, or SPTB, as women without HIV. And in Lusaka, Zambia, the estimates vary. It's actually we're actually a few years behind at this point, so I'll be looking for the next report, but up to about 20% of pregnant women are living with HIV. Yet the underlying immunological mechanisms of spontaneous preterm birth and its association with HIV infection are not known. And then I think it goes without saying, I think this group all appreciates this sentiment, but in Zambia and many other limited resource settings around the world, including in some parts of our own country in the US, a diagnostic method to predict possible adverse birth outcomes could greatly improve the health of infants and mothers. We have limited resources in many places globally, and being able to identify a woman who might be at increased risk of having a spontaneous preterm birth or some other adverse birth outcome could really help us direct the resources we have to helping those most at need. So we were interested in evaluating S1P as a potential predictive marker for preterm birth. And if it turned out not to be, at least evaluating the technology and the sort of proof of concept experiments to see whether this is a concept that we could employ in the future, either with S1P signaling or some other biomarker. So again, this is the same figure that you've seen before, but sort of the thought process behind this, like what might be the connection? Well, we know that HIV is known to cause inflammation, and this alters S1P signaling in immune cells. S1P changes are also observed during pregnancy, and I'm going to show you one study that looked at that a while back. The research is very limited. And again, in turn, blocking S1P reduces HIV infection, which is what I've shown you previously. So we hypothesized that there may be some interplay between HIV infection, S1P signaling, and pregnancy that could influence pregnancy outcomes. And therefore, I hypothesized that S1P levels may differ by HIV status and birth outcome. So I mentioned that there is very limited literature about S1P signaling and female reproductive immunology, and I generally make an effort to check back on this regularly, so I have to look back in case anything has come out in the last several months. But as of recently, it's really very limited. There's a lot to learn. In 2015, Rie et al. Showed that alterations in S1P signaling are observed in cases of pelvic organ prolapse. So in a case control study, they showed that in human vaginal wall specimens, S1PR 1, 2, and 3 were increased in cases of POP versus control. And they discussed how S1P is a known regulator of smooth muscle contraction. This is one of its many functions, like I talked about at the start. So this could be a possible mechanism of action. And then Lee et al. Showed that in polycystic ovarian syndrome, or PCOS, S1P, ceramide, and sphingomyelin, which is part of the S1P biosynthesis pathway, is increased in PCOS. So PCOS is now I'm speaking to the biomedical scientists, you're probably familiar, but this is a can be very devastating condition for women because it causes a lot of symptoms, can interrupt the quality of life, and can also hinder fertility. So they thought that this might be one of the S1P signaling components, might be a potential biomarker or diagnostic, which is really exciting to me because this is something I wanted to look at, but so far I haven't seen their follow-up publications on S1P and PCOS. And then finally, quite some time ago now, Yamamoto et al. Showed that modulation of S1P signaling occurs in the decidua of the placenta leading up to birth. And I'm not an expert at all, but I understand this to be a layer or part of the placenta. Anyone, please correct me. But it was really cool what they saw. They actually looked at increasing gestational age, and they saw increasing sphingosine kinase protein levels and activity, which were positively correlated in the placenta leading up to term birth. And they also saw that S1P lyase, which is an indicator of the turnover of S1P, was increased at term. And then lastly, S1PR3, so that receptor 3 was also increased. And this is particularly interesting to me, which will become apparent in the next couple of slides. So we know that HIV causes inflammation. This is a very general potential causal diagram. And that seems to be an influencer of S1P levels based on some of my earlier work in the human thymus. We also know that there's some relationship between HIV inflammation and preterm birth. This is something that my current organization, UNC at Chapel Hill, is really interested in. They're doing a lot of work on soluble immune markers, multi-omics analyses, inflammation, things like that. What we don't know is if there's any relationship at all between S1P levels and preterm birth. This was something that was not looked at, apart from that paper in the placenta that I mentioned, where they showed increasing S1P signaling leading up to or with increasing gestational age. So this was unknown, and I was really curious whether there may be some triangulation between the three. But first of all, we wanted to see, is there a relationship between S1P levels and birth outcome? So this is the title of my proposal. And again, my main hypothesis was sphingosine 1-phosphate signaling in the reproductive tract of women with HIV may differ from that of HIV negative women, and this disparity may be associated with preterm birth. My main objective, and what I've really focused my time on since being here, is to quantify the level of S1P signaling in the female reproductive tract and investigate whether it's associated with HIV or spontaneous preterm birth. In the future, I hope to integrate some of this data with ongoing multi-omics and microbiome analyses that are ongoing at UNC, as I mentioned, and then a personal interest that I'll look at if time permits and if my STATA skills evolve a little beyond what they're currently at, will be to explore the socioeconomic and other epidemiologic characteristics or clinical data associated with an inflammatory female reproductive tract environment, S1P signaling, HIV, and adverse birth outcomes. So I really think it's that we need to take a really big picture approach. But so far, what I've focused on and what has really just taken the most of my time here is this aim one of the project. So this is the study design. So as I mentioned, in Lusaka, Zambia, we have this ongoing longitudinal antenatal cohort, and I was really privileged to come into this this group where this study has been established since 2015. So we call it ZAPS, although now we are collaborating with Gates, and it's sort of taken on the name Prisma. And Emily Smith from GW is actually leading this. So I've really had the opportunity to continue to work with GW colleagues. ZAPS, as I mentioned, is an ongoing longitudinal antenatal cohort based here in Zambia. It's also known as the Zambian Preterm Birth Prevention Study, or now PRISMA. We enroll women in early pregnancy and follow them through delivery and the postpartum period. And then at multiple time points throughout the pregnancy, they come in for clinical visits where they have a clinical check, and dry vaginal swabs are collected by research nurses via speculum exam and stored at -80 in our on-site biorepository. And I should mention that during those visits, it's not only the vaginal swabs that are collected, but also blood and biospecimens, which are used for really a myriad of studies that are ongoing here. I see there's a comment, so I'm going to, oh, thank you, Natalia. I just wanted to check if it was the question. I don't mind being interrupted. So this was a pilot study, so it was a relatively small scale. We had 160 specimens to start with, and there were 40 swabs from each group. So we looked at HIV negative, full-term birth, HIV negative, spontaneous preterm birth. And I should just mention that means that the preterm birth was not in any way provider initiated. So it was just they weren't induced or anything. It was just a preterm birth that just happened. HIV positive full-term birth and HIV positive spontaneous preterm birth. And I evaluated in those swabs the expression of S1P receptors 1 and 3 and sphingosine kinases 1 and 2. So our main outcome of interest, again, was spontaneous preterm birth. And this, for the purpose of this study, we define this as fewer than 37 weeks of gestation. Our main predictors and covariates, the big one was the S1P biomarkers as well as HIV infection. So we have data on viral load as well as CD4 levels and ART status. And then, as I mentioned, in the future, I'm really interested in looking at some of these other clinical factors and epidemiologic factors like perceived stress, SES, sanitation, employment, et cetera. And we have all of this information, which is gathered and integrated from household surveys of participants in the study. And we also excluded twin or greater gestation. I just asked somebody the other day, actually, because the form, when we women consent to the study, to enter the study, is there's a section that indicates, is there a triplet gestation or 4 or greater? And they said that we don't really see that in Zambia. It's very infrequent, but in some of the other study sites that we're working with, like in Asia, they do tend to have sometimes triplets or more. So in the lab, I quantified the expression of S1P receptors 1 and 3 and sphingosine kinases 1 and 2 in those stored frozen vaginal swab specimens. This was done by RNA isolation, and I just did this with a good old QIAGEN RNA-Z extraction kit with an additional gDNA removal step, and it worked very well to really just clean everything up. And then quantitative real-time RT-PCR using the housekeeping gene GAPDH as the control. So we looked at the relative expression of all of these markers to the housekeeping gene. And then the first thing I wanted to look at, and the first data I wanted to present, was just to describe the distribution of markers by birth outcome and HIV status, again, in those 4 groups. And then I've done some basic 2-sample t-tests for paired group comparisons. And then in the future, we'll do some further analyses like ANOVA, logistic regression, et cetera. So I was really excited to see, when I started getting my data back, that we actually do detect components of S1P signaling in these dry vaginal swabs from the female reproductive tract. And we were able to detect both of the sphingosine kinases as well as 2 of the receptors. And that's all I was able to look at so far with my fellowship budget and time. But it was still exciting because, as I mentioned, this is really an understudied area and had not really been done by PCR from vaginal swabs. And so we saw that looking at the kinases, there was really comparable expression of sphingosine kinase 1 in all women. There was possibly a slightly higher average expression. This is just, sorry to skim over this. This is just showing the expression of the target relative to the housekeeping gene. This is just the way that we analyze that. There was slightly higher expression in women living with HIV, but really pretty much the same across the board. And then when looking at the different groups according to HIV status and birth outcome, we saw, again, really no difference in the kinase expression across all 4 groups. And the same was true for sphingosine kinase 2. We saw slightly higher expression, non-statistically significant, we checked, in HIV positive women versus HIV negative. And then looking across the 4 groups by HIV status and birth outcome, pretty comparable expression. Interestingly, we saw a lot of variability in the HIV positive preterm birth group. And just make a note of that because we actually saw the same for the receptor expression. So this particular group seems to have a lot of donor variability. And my hypothesis, although I haven't been able to look at that yet, is that it may be due in part to the CD4 levels or something else related to infection, like perhaps the viral load. Although I should also mention that all women in our study are on ART, they are provided ART. However, we don't necessarily have an optimal way of checking, the fidelity with which that's being taken. But theoretically, all women are on ART. But I thought that variability might have something to do with HIV infection. Then things became a little more exciting when we looked at the receptors because we saw that there was now definitely a trend of increased expression with S1P receptor 1 in the HIV positive participants relative to the HIV negative. And then when we looked at S1P receptor 3, there was definitely increased expression in women living with HIV versus the HIV negative participants, which was statistically significant. And then there's a mathematical formula where you can calculate the fold increase from PCR data. And this translated to about a 2-fold increase and this a 3-fold increase in S1PR3. Then looking across the 4 groups of interest, again, at S1PR1 and S1PR3, we saw that there were a few different variations of notes. So with the HIV positive preterm birth, we saw increased expression versus the HIV positive full term birth, not significant though. And then we saw increased expression relative to the HIV negative preterm birth. So holding the birth status constant, so among all the women with preterm birth, those living with HIV had higher S1P receptor 1 expression, and that was statistically significant. And then looking at S1PR3, again, we did see some increases, and specifically in the HIV positive full-term birth versus the HIV negative full-term birth, we saw a statistically significant increase. So holding the birth outcome steady, so all of the full-term birth individuals, those living with HIV had higher expression of this receptor. And then when comparing the among all HIV negative women, those experiencing preterm birth, so the SPTB versus the full-term birth, also had higher S1PR3 expression. And that was also a greater fold increase than what we had seen previously. So I'd like to mention some of the strengths and limitations of this this is very much a pilot study in all senses, in all sense of the term. Its strengths are primarily the fact that it's embedded in a large, well-characterized parent study population. So as I mentioned, ZAPS and now PRISMA has been ongoing since 2015. We have a very high retention and follow-up rate, which is great. And as I said, we involve the women throughout the pregnancy and then postpartum as well. So really a wealth of biomedical data, clinical data, extensive epi data is collected, as I mentioned, in both in the clinic and in household surveys. And also the swab collection it's really well established and it's actually acceptable to the women. So one nice comment that I heard from one of the nurses who I was chatting with was that her friend, who's about 40 years old, found out that she's pregnant, and she enrolled in our study. And she mentioned that she's really glad to have the chance to be in the study because she comes in at these regular intervals throughout the pregnancy. And even though she has to have a swab collected, she doesn't find it to be a big deal because she's glad that they're checking at those regular intervals. And if anything comes up, they're going to catch it right away. So she considers, because of her age, she thinks maybe she's a little bit higher risk. And it really gives her sort of a sense of peace because this large number of visits tends to be more than many women in Lusaka do go in for as antenatal care. Also, the qPCR testing is fast and cost effective. In my project, I looked at S1P by qPCR, but this would theoretically be possible for a wide range of immune markers or other parameters. So this proof of concept of RNA extraction followed by PCR is promising. This is something that can be done in about a day. Some of the limitations are that this is an observational study with a small sample size. And so I can't establish any causal relationships. It wasn't designed or powered for that, but it's a good first step. So in the future, I would love to characterize the expression of the other S1P receptors. I have not yet been able to optimize the extraction to obtain sufficient RNA where I can do everything that I want to do from one specimen, which is unfortunate. I was only able to look at 2 of the receptors and the 2 kinases, but in the future, it would be great to also see what's going on with these other receptors, because who knows, it might be even a bigger difference than what we've seen with S1PR1 and 3. And then another idea that I had, which would be really cool, is to look at S1P signaling in PBMC collected from the same donors that we're looking at the vaginal swabs from. So sort of sister samples, as I've heard them referred to as. And this would be really interesting because if we see any difference in the vaginal swabs, we could possibly relate that to some difference that we also see in the PBMC specimens. So for analysis, as I mentioned, I have a lot of covariates of interest, the main ones being the parameters related to HIV, like time of ART initiation, CD4 levels, and then looking at the association with viral load and CD4 count in women with HIV might contribute to our understanding of some of that variation that we saw with the HIV positive preterm birth. And we also saw that, I'll just quickly hop back, we saw that really notably with S1P receptor 3, where we had some of these individuals with very low expression relative to the housekeeping gene versus, some of these ladies had really high expression. So I'm really curious mechanistically what's going on here. Like, maybe some of them have a less controlled virus, and that's contributing to greater inflammation, which even though they are all on ART, we know that there's still some viral shedding that may occur in the reproductive tract. And maybe that could be contributing to increased localized inflammation, which could then be causing cytokine changes and S1P receptor changes. So you can tell that I'm still interested in basic science. So these are just some analyses that I'd like to do. Again, bringing in all of the epi data that we have and looking at any big picture questions between systemic and local inflammation, perceived stress. So really tying in the big picture here, as well as working with UNC on the multi-omics and microbiome studies, to the extent that it's possible in the short time that I have left. So one example of an analysis that I hope to do in the future is looking at the outcome of preterm birth. Logistic regression analysis could incorporate all of these predictor variables, like the S1P biomarkers, viral load, and CD4 count. These are just the types of variables. Perceived stress level, HIV infection, etc. So just to summarize, the S1P receptors and kinases are expressed in the female reproductive tract, are detectable by RNA extraction and qRT-PCR, and they differ in our small study by HIV status and pregnancy outcome. Sphingosine kinase 1 and 2 are comparable among all of the groups that we looked at, but the receptors are increased in HIV positive women and in preterm versus full-term birth among HIV positive women, and that's for S1PR1. Then for S1PR3, we saw an increase in preterm birth versus full-term birth among HIV negative women. So I believe that these studies suggest that further investigation of S1P in pregnancy, especially among women living with HIV, is warranted. And just to tie it all back together, I'm really excited to have had the opportunity to look at evaluating a certain type of testing for a biomarker that may possibly contribute in the future to earlier identification of women at risk for an adverse birth outcome. Now, I don't think it's going to be S1P. I'm willing to admit that. It's probably not our panacea, but I'm excited to have provided this proof of concept that in our vaginal swabs, we can easily extract genetic material and do PCR to test a variety of biomarkers. So I would just like to thank, first of all of the ZAPS and PRISMA participants, the families and children. I don't know if this is going to play, but these are those guys again, they're back with their little sticks. And then here we are on a hot day. Sorry for the noise, it's a little loud.
We can't hear it.
We can't hear anything.
Oh. I don't know if you saw it play, but they're just screaming with joy as they run.
They look happy.
Yeah. And then I would like to thank all the individuals who have mentored, who are listed here at UNC, GW, Hopkins, and in Zambia especially. And my funding is here. So thank you very much. I know I went a little bit long, but I'm happy to take any questions.
No, you're fine. Thank you so much. That was a fantastic talk. I have about 100 questions for you.
Me too.
But I'll let Martha go first.
Oh,
I don't know if I should have stopped sharing. I'm sorry. I can bring it back.
No, you're fine. You're good. You're good. And I have to run in at exactly 3 o'clock because I have another call. So Vivian, I'll have you take over for me if you guys are still chatting. And Rachel, if I don't have time to chat with you, I would love to set up another conversation. I have lots of thoughts and ideas.
That's really exciting. I love that. Thank you.
Yeah, and if I can be part of that too, but I think you kind of answered my question in the summary. And this talk is just so close to my heart because I'm from Zambia and I've had two babies. And remember, we met right before you were going. And both of them have been preterm. Both babies were preterm. And often wondered, like they didn't want me to go into labor, obviously they wanted me to have a C-section and so on. But I wanted to tie it all together and I didn't seem to have heard. Is what you're saying that the biomarkers, finding those biomarkers can help develop into something that can help prevent preterm? Is that what we're trying to do here? I listen to everything, but some of this scientific stuff throws us off.
I'm sorry, it was a lot of basic science. That's just where I come from, but I keep trying to learn how to communicate it in a universally understandable way. Well, first of all, thank you, Martha. I know we talked before and you made me feel really excited about going to Zambia. And I can only say that since I've been here, the warmth of the people and everyone I've worked with has just reinforced that over and over again. So thank you for coming. And to answer your question, with my limited time that I had here and the funding I had, I wasn't interested, not to say not interested, I wasn't in a position to start looking at something that could be preventative. I was more interested in, first of all, just from my personal interest, if the signaling pathway was active in the female reproductive tract, and then if it could possibly be like a marker for catching a woman who might be at risk for having a preterm birth. Because if we see differences like in the preterm birth group, for example, if I would have seen super high S1P levels, then maybe we're testing women, we can test that as they're coming in for the visits and we can say, this woman already has really high S1P levels. We need to keep an eye on her and direct the resources we have to help, because she may have a preterm birth. But we do have a lot of clinicians who are working here as part of ZAPS, now PRISMA, and they're doing a lot of other things trying to address something that might be preventative, but I haven't had the opportunity to do that.
Okay.
So
thank you.
I hope that helps clarify a little bit.
To build on Martha's comment, and then I know there's other hands raised, I want to just make the point that the variability in preterm birth, I wouldn't find in terms of your S1P data, I wouldn't find that discouraging, especially in small numbers, given how multifactorial preterm birth can be. I mean, I'm an obstetrician for context, but yes, it may well have something to do with the S1P pathway, but it may also be completely independent. And you can't assume that that inflammatory pathway is necessarily going to be relevant in all etiologies of preterm delivery. We do see a lot of preterm delivery at our hospital. I was just looking at our cohort data. We're at about 20% for women with HIV, about 12% for propensity score matched controls. And it holds true with looking at a spontaneous versus iatrogenic. But I was wondering, the other outcome that we find most notably, at least in the US, is small for gestational age among birthing individuals with HIV, and which likely also has some relationship with inflammation, although that's pretty unclear. So it may be interesting, just as a secondary analysis at some point, to take a look at what those birth weights are by percentile, if you have them, by gestational age, and see how that data plays out, and see if you see the same correlations. Because I have a sneaking suspicion that they may also align more than you necessarily would have expected. I think my last question actually was from about halfway through. Those were comments, so my apologies. But
no, thank you very much.
Love to discuss more. As a very enthusiastic obstetrician, I would be more than happy to discuss at a later time. But I guess my question was in terms of longer term clinical application beyond biomarkers. Would you be looking at like an S1P receptor modulator, or, you know, in terms of like an intervention that would potentially not just identify, because when we identify preterm delivery or you know, women at risk for preterm delivery or low growth restriction, there's not much we can do as obstetricians. But you know, would that be, I don't know much about S1P receptor modulators or modulators in general, or those modulators in general. So I was just wondering what your thoughts are in terms of potential eventual clinical application. And I'm sorry, I actually need to run, but that was my question.
Oh, that's okay. We can talk more about it later too, but if you have to run, it's fine, but I'll just answer that to the best of my ability. So we thought about that actually with Alberto when I was looking at HIV in primary cells, and the idea was there could be a possible clinical implication at some point in the future, because this was established for MS. This was already being used.
I vaguely remember that from MS.
Yeah. However, in the context of pregnancy, I think it would be really complicated because there are, you know, S1P expression is ubiquitous, and I'm not sure kind of what would be the mode of delivery, you know, if it would be something topical or systemic. I have very limited clinical.
Targeted, yeah.
Right. It would have to be, exactly. It would have to be very targeted in order, I think, to be used in that context, just because of the ubiquitous nature of S1P signaling. Yeah.
All right, well, let's have a longer conversation, and I will let Vivian moderate. My apologies that I have to run, but I'm already late. Thank you so much for your talk.
Thank you. Thank you very much.
Thanks, Rachel. I know we're past 3 p.m., but I see a few hands up. Dr. Liu, would you like to ask your question? If anyone, you know, has to drop off, of course, I understand.
Thank you. Hi, Rachel. This is Dr. Ben Liu, Assistant Director of Microbiology at Children's National Hospital. So I have two questions. The first one is about your basic science research. For the if you look at your flow cytometry experiment, when the primary CD4 cells treated with the FTY inhibitor, seems if you increase the concentration of FTY, the inhibition of P24 expression cannot be reached to the zero. Seems that there's still some residual P24 expression there. Looking at your cartoon, seems the SP1 can give some influence on the SP1R. I wonder if you or Alberto has done experiment in some cell lines without the feedback of SP1, S1P, to see whether without the S1P, the inhibition of P24 can be down to 0%? This is my first question.
Mhm. Yeah, that's a very good question. Thank you so much. We haven't done experiments in cell lines. Actually, I only worked exclusively with primary cells in Alberto's lab. And actually, even before that, when I started out looking at S1P, it was in human thymus specimens. So it's always been primary cells. And I think we've always kind of preferred that just because we believe it to be much more physiological. But I think from a mechanistic point of view, just getting to the mechanisms, I appreciate your suggestion. And I think that would be a good experiment to do.
My second
But I
question,
haven't done it.
Yeah, thank you. Given I'm a laboratorian and working with the different biomarkers in terms of predicting or diagnosing infections, I wonder if I could ask some question on your second project. The vaginal swab is a non-sterile source because the bacteria can also express S1P. So you don't know whether the S1P you detected is due to bacteria or due to human expression. I think detecting S1P from sterile source like a plasma or blood may give you a clear background. The second one is, the S1P, the correlation between S1P may not be just a positive and negative with the preterm birth. I think developing a quantitative PCR to determine the quantity of S1P to perform the raw curve analysis can give you a threshold to correlate with the level of S1P and the risk of preterm birth.
Yeah, thank you so much. So for your first question or point about the vaginal swabs, they're definitely not you're correct about that. It's not a sterile source. And I was also really curious, and I see Christina here on the call, my mentor from UNC. We've talked about this a couple of times about the concept of what is going on with the vaginal microbiome, and is there S1P, the actual ligand, just clarifying, is there S1P present that is produced by vaginal bacteria? And I'm sure that there probably is. So for these PCRs, we did look at we used human-specific primers for the receptors. So they should be specific to human S1P receptor 1 and 3, as well as the kinases. But of course, it's always good to check that. So I really appreciate that point. Yeah. And about your first point as well, there are some really cool papers showing that, the S1P produced by bacteria in the gut, for example, can enter the human metabolism, and we can actually metabolize that. So it's just really crazy, crazy to think about. And then I didn't fully hear your second point. Because there was a notification, but I think you were talking about developing a quantitative PCR assay versus a relative expression. And I think that's, that's a great point. Yeah.
Thank you.
Thank you so much. Mia, can I take your question?
Go ahead.
Sorry, Vivian.
Sorry, my name is Mia Kamara. I'm an undergraduate student at Howard University, and I'd just like to say I was really intrigued by your presentation and the topic that you were speaking on. That's just a little bit close to home, as I am a first-generation student from Sierra Leone, which has a large population of women that live with HIV. So my question for you is like, with your research project and with this research, is there opportunities for undergraduate students? And I would just love an opportunity to connect with you further on this topic and talk to you a little bit more about it.
I would be more than happy to talk with you. I'm not sure about specific undergrad opportunities, but I know that the fellowship I applied for has sort of a postdoc fellowship and a earlier career fellowship component. So you might be interested in something like that, but I'll think about it some more, and I'd love to connect. I can put my email in the chat. And thank you so much for coming.
Yes, thank you so much.
Are there any other questions?
I see Taz. Taz, it's like 10 p.m. in Zambia. I can't believe you're here. Thank you.
Sure. That's clear.
Well, I want to thank everyone so much. I know I went a little over. If there's any more questions, I'm more than happy to take them.
Great, and thank you, Rachel, for adding your email to the chat. If there are no other questions, I did want to thank everyone for joining the meeting and Rachel for giving such an excellent talk. It seems we should have scheduled this call for maybe 90 minutes so that everyone could have answered their question or asked their questions and had them answered. But as Rachel Scott had mentioned, we'll be following up with the recording of this session if you'd like to look back at it in the coming days. Yes, thank you so much for joining.
Thank you, Vivian. I don't know if there's any way to save or export the chat. I see some people made some really helpful comments, but I'm not able to.
Yes.
Maybe I can take a screenshot if you can't export it.
I can definitely, I think I'll have a recording of that as well. So I can send that to you as well.
Thank you.
Thank you all.